Hi, so for this video I'll explain what's going on while we extract DNA. So for today's experiment we're doing DNA extraction. So I'm just explaining what's going on step by step when we do the experiment. And this works with any plant cell, animal cell. Most commonly is known for the kiwi cells we use, onions, banana works as well. So we try that out. And here's my nucleus. I'm put them here, here. It's basically stacking up all the plant cells together. Okay. And the first thing we do is that we mash it up in a mortar and pestle. Okay. By doing that, we physically are tearing up the cells, freeing them up from each other, making a big mulch. And this would increase the surface area for the cell's membrane to the solution. And the solution we're putting into is called the extraction buffer. Extraction buffer. Okay? And inside that buffer we have hand soap, which is also known as a detergent. And the detergent has a common feature with the phospholipids, the cell membrane, which is they are both hydrophobic. So when they're both hydrophobic, they tend to group together. And he would just go in and it's like, "Oh, what's up? I'll hang out with you guys in the cell membranes," and just hang out there and just wedge themselves there, space by space, and kind of breaking them apart, intruding their space. And then the salt has another effect. It creates an osmosis gradient because water will go from a lower concentration of salt to a high concentration of salt. So it will push outwards, and by doing this push, it will lyse the cell mechanically. So we will break the outer cell because the water is pushing it away. And this will free our nucleus. Okay, I'll draw the nucleus here. And then same thing happens with the nucleus as well, because the nucleus has the same features as the cell membrane. The membrane from the outside to the inside. So this similar thing happens. The detergent come also around, splitting up the phospholipids, and salt water creates a osmosis gradient, osmosis pressure, breaking out the nucleus. And inside that nucleus we have our good old-fashioned DNA. Yay! Okay, now the next step of our DNA extraction is putting it in some alcohol. Now what happens that after we filtered the solution, we end up with some kind of like a liquid goop or a liquid kind of thing coming down, and DNA is dissolved in that liquid. Okay, it's something that cannot really be seen or touched. So what we want to have a physical DNA floating around, so we can actually look at it and see it, and see what it looks like. Fortunately, that it could happen. We could precipitate DNA by adding ethanol. Okay, precipitate means forming a liquid. Well, not liquid, forming something that's in a liquid form. Not in liquid form. Sorry, I'm using the wrong terms here. Precipitating means that we have something in liquid, in solution. That's a solute. There you go. And we'll just bring it out to solid. So when DNA is in contact with the ethanol, it will precipitate, and you have this nice stringy stuff between the two layers of the alcohol and the fruit mush at the bottom of the beaker. And from there you can just take your fancy homemade hook, bring it down there, and fish up your DNA. And that's how we extract DNA. That's about it. See you next time.